So thanks very much, Florian. So today our task is to review the findings of our combined HBSL and LBSL work group. And so first of all, I'll basically be giving the introduction and Heatmap overview, followed, and I'll then talk more specifically actually about the key successes and challenges in HBSL. Then Beth McGinn will come up from Cure LBSL to talk about, to focus on a couple of the successes with LBSL, and Dr. Christina Nemeth from Kennedy Krieger about key challenges. And then hopefully we'll have some time for discussion and questions. And I just wanted to say thanks again to Adeline and Omar and other GLIA leadership for organizing this meeting and for including us at, these two very ultra-rare leukodystrophies this year. Okay, so thanks again to our work group. We had a pretty large group between the two disorders. I just wanted especially to give thanks to our HBSL crew who contribute a lot in terms of edits and additions to the rubrics. And specifically just wanted to mention Stephen Damiani of Mission Massimo, Dr. Chloe Stutter, Dr. Dominic Froelich, Dr. Matthias Klugman, and Dr. Mohammed Shaker. Okay, so just to review for those who might not be as familiar with these two disorders, HBSL is hypomyelination with brainstem and spinal cord involvement in leg spasticity. And there are DARS mutations in that disorder. And then LBSL, leukoencephalopathy with brainstem and spinal cord involvement and lactate elevation. So I'll try to explain some of the rationale as to why these were included in the same work group. So these are related leukodystrophies. They comprise a phenotypic spectrum with overlapping MRI findings in terms of the distribution. So there are white matter changes in the cerebral white matter, as well as cerebellar, brainstem, and spinal cord white matter tracts. In HBSL, there's hypomyelination versus in LBSL, the features are multifocal T2 signal hyperintensities. So these are tRNA synthetase disorders. I'll explain a little more about that, but they belong to different cellular compartments. DARS, or DARS1 is cytoplasmic, DARS2 is mitochondrial. So in both cases, these enzymes encode aspartyl tRNA synthetase, which is an enzyme that's critically important in attaching aspartate to its cognate tRNA and the process of amino acylation. And so it's also, though, well known that these enzymes also play non-canonical roles in terms of cell signaling and immune regulation. So that's contributing to the complexity. They're both autosomal recessive inheritance with usually compound heterozygous or homozygous mutations in either DARS or DARS2. And then in terms of the clinical presentation and how early symptoms present, HBSL usually presents pretty early in infancy or early childhood, actually most patients before 1 year of age. And LBSL, there's really a very wide spectrum. So some patients actually have symptoms as soon as they're born, sometimes infancy, childhood, or even onset as late as adulthood. And that spectrum has particularly expanded in the last 10 years. So we know that there's, as with many of the other disorders presented today, quite a variable progression rate, but one trend that seems clear is that earlier onset of symptoms tends to be associated with greater severity and more rapid progression, unfortunately. In terms of the clinical findings, more specifically, there are progressive neurologic findings that reflect involvement of the brain areas that I already mentioned. So pyramidal tracts, especially in HBSL, there's spasticity, brisk deep tendon reflexes, weakness. In the cerebellum, there's ataxia, especially affecting the lower extremities, tremor, dysmetria, nystagmus, and then the dorsal columns are involved, really universally with sensory ataxia and falls being common. So cognition is variably affected. It's affected less often than motor function. There's cognition problems or even intellectual disability more commonly in HBSL. Although having some developmental delays, learning disabilities, attention issues that require school support, that's often reported in LBSL as well. Okay, So I'll just first start with an overview of our research challenges and successes in LBSL, or DARS2-related leukodystrophy. So as this just really echoing what others have said today, which is that the advocacy groups really have been the backbone of support in LBSL. A Cure for LE, or soon to be named Cure LBSL, has been a major driving force in terms of fundraising and supporting patient engagement. The co-founders are Beth and Mike McGinn. And as I mentioned, they really have heavily supported our clinical and basic science research efforts through fundraising and helping to recruit patients for our clinical studies in natural history. Cure LBSL is currently establishing a global LBSL research consortium and a formal patient registry now through the Chan Zuckerberg Institute grant, which they were just awarded in the last year. We're very excited about that. That'll be $600,000 of funding over the next 3 years for these initiatives. And also excited to anticipate patient listening sessions with the FDA upcoming, hopefully in spring 2023. So another success that we consider in LBSL is that the wide phenotypic spectrum is pretty well established in the literature. I already alluded to the fact that there's clinical heterogeneity, and this is very rare. We know of maybe 200 cases worldwide, so that will continue to complicate clinical trial design. However, to try to address this, the LBSL community is building approaches to engage more patients in international sites, including remote assessment validation. So as an example, we do have natural history studies underway in the US at Kennedy Krieger since 2018, and in Amsterdam at Amsterdam University Medical Center since 2020. And there's also a site in Finland that is upcoming. They're just awaiting their institutional review board approval. And so we're working together. We're really collaborating. We have standardized protocols across these sites, which should help facilitate the process of getting regulatory approval. And we're collecting a battery of different tests, so ranging from clinical and neurological information, neuroimaging data, and then also quantitative sensory motor outcome measures that capture detailed information about walking and balance impairment in patients using wearable sensors. So next, another advantage is that we have a great diversity of preclinical models to address questions about disease mechanisms and therapeutic targets in LBSL. As others have mentioned, one drawback is that none of these models accurately does recapitulate all aspects of the human disease, but we have several mouse models, cerebral organoid models, which are more recent, and as well a patient iPSC and neuronal studies ongoing. So despite these efforts, we still do have a limited understanding of disease pathways, and that's a clear target for disease-modifying therapy, so that's one drawback, but Dr. Mertz will talk a little bit more about the fine points of that. Okay, and then finally, we still do lack blood and imaging biomarkers to assess disease severity and monitor progression, but this is also something that we're working on. So as one example, we're looking at advanced imaging sequences of the white matter and the brain and the spinal cord to try to determine if there are any MRI biomarkers we can use going forward. Okay. So in terms of clinical challenges and successes, there were two major ones that our group agreed upon. First, that there's really a well-established diagnostic approach consisting of the MRI pattern, clinical criteria, and genetic testing. And Dr. Mario Vandernap's group was largely responsible for this going back about 20 years ago. Unfortunately, though, the success of this approach is based on experts only available at limited centers, and we still have barriers to genetic testing, access and affordability in countries with limited resources, which is, of course, a common problem. At this point, we did agree that early diagnosis through a mechanism such as universal newborn screening, unfortunately, is likely not feasible. And then another point is that even though there's really a nice body of literature over the last 20 years and ongoing natural history studies, we haven't made a targeted effort to develop guidelines for symptomatic care in LBSL that can be published as an international standard. But this was proposed, and we look forward to continuing that work coming out of the work groups. Okay, so just to touch quickly on the LBSL Heatmap overview, in the disease-modifying pathway, we really have made the most advances in terms of preclinical scientific development and advocacy, as well as outcomes and clinical planning. But still in the red is therapeutic development, and sort of our next immediate step towards that is the patient-focused drug development meeting that we're arranging with the FDA through with Cure LBSL, hopefully in 2023. And then in the disease-specific clinical pathway, really the most progress has been made in understanding symptoms and symptomatic care. But as I mentioned, one immediate goal that should come out of our work group is that we can further optimize this by establishing international care guidelines and publishing those. Okay. So then I'll switch gears now to research challenges and successes for HBSL. And so Massimo's mission is the primary advocacy organization for HBSL. They've been very active and productive in the last 10 years. The co-founders are Stephen Damiani and Sally Damiani. And generally their mission, this is a clinical and research program that promotes prevention, diagnosis, and treatment of childhood LDs. Really, HBSL is intended to be the prototype for other rare disorders, and they hope that it'll be a successful model to use that translates to other leukodystrophies. They've helped to recruit scientists and clinicians that have advanced preclinical research studies in HBSL since 2013. They've contributed to awareness of HBSL through media outreach, and they're partnered with broader leukodystrophy patient and family support organizations. This is important, especially because I should mention that this HBSL is even more rare, first described only about 10 years ago, and we're aware of about 20 patients worldwide. So, so there's work to be done in terms of identifying other patients, and then also, trying to join with these larger groups to push the mission forward. Another advantage in HBSL is that we do also have a great diversity of preclinical models to address questions about disease mechanisms and targets, also an area that's rapidly advanced in the last 10 years. Some of the more recent models just in the last 2 years have incorporated patient-specific mutations, but as is the story in LBSL, we still lack a model that more fully recapitulates the clinical aspects of the disease. There's not a clear target identified for DMTs yet, except gene therapy, with this being a monogenic disorder. And there's actually no central registry or ongoing natural history study yet. We think most likely not initiated due to the ultra rarity. But we, of course, need this to further expand the phenotypic spectrum and identify outcome measures that are important to patients. So in terms of clinical challenges and successes, there is a growing number of patient support organizations that are either specific to or that include HBSL, including Leukodystrophy Australia, Yasha's Foundation in Amsterdam, and we hope that through the participation in these organizations, that might help facilitate enrollment when we do start a registry or clinical research studies. Genetic testing is the main barrier really to confirming an HBSL diagnosis in locations with limited resources. But one group that's made a difference is the Illumina I Hope program, begun in 2020, which is aiming to make whole genome sequencing accessible to underserved children who have suspected genetic diseases. And then another point that was brought up during the work group meeting is that there have been some preliminary talks with Dr. Mario Vandernap's group to try to establish a consistent pathway for functional analysis of US as an HBSL. As I mentioned in LBSL as well, unfortunately, early diagnosis through a mechanism such as newborn screening is likely not feasible either at this time, and we don't have established or published clinical care guidelines yet, but there was a lot of interest generated in this during the work group. Okay, so just to kind of give the overview of where we are with the Heatmap, our very first Heatmap, which we also hope over the next couple of years will turn more to greens and blues. So really the strongest areas are preclinical scientific development and advocacy. In terms of the outcomes in clinical planning, really that has to start with getting the patient registry and natural history ongoing. And so we think that one way we can do this really is to leverage the experience of the LBSL advocates and investigators to get this process going. And then in terms of the disease-specific clinical pathway, really more oranges and reds here, but at least in terms of symptomatic care where there's the most progress, we agreed that working on clinical guidelines that can be published is a good next step. Okay, so over the next few slides, I'll just go into a few more specifics about successes and challenges in HBSL. So again, I already mentioned the advocacy work of Massimo's missions based in Australia, and they're really broadly trying to improve diagnostic yield and scientific research in childhood leukodystrophies with HBSL as the prototype. They've been collaborating with scientists to advance preclinical research. They've partnered with those broader leukodystrophy organizations I've mentioned, and they're very actively engaged in mass media and social media to raise awareness. They have advocated for federal government support of this research and raised $3 million over 5 years, and they hold an annual research update conference every year since 2020. So another major success really is the diverse platform of preclinical models to help understand mechanisms of disease. So 2 of these are iPSC and CRISPR mouse models that have explored the functional effects of DARS1 mutations. And so the summary of what's known so far is that these mutations do impair aminoacylation, but secondary ARS functions might also play a role. They've established that neurons do have a higher aspartyl-tRNA synthetase expression versus other brain cell types, both in the mouse and human brain, and the very strongest expression is in the cerebellum. Doing a full DARS1 knockout is embryonically lethal in mice, but really just in the last couple of years, there are a couple of viable DARS1 models that have been published that introduce patient-specific point mutations, and these do look more closely parallel the human condition with developmental delay, early death, and then those animals that survive have later onset motor deficits and spinal cord white matter changes. So in terms of sort of the final line of research that's more recent is to establish oligodendrocyte brain organoid models. This has been work done at the University of Queensland by the Wolvetang lab. And so through these mouse models, or through these organoid models, they have found that treatment with LOLA to boost aspartyl-tRNA synthetase, increase oligodendrocytes and organoids, and then treatment with lysolecithin, as expected, was able to cause cell death and induce hypomyelination. So this appears to be an effective model for drug screening in HBSL. Okay, and then just one more slide on key challenges that our work group identified in HBSL. So first, as I already alluded to, there's a need for a model that more faithfully recapitulates the clinical aspects of the disease. We don't yet have a clear target for DMTs, and potential targets have been discussed but really not validated, such as gene therapy. And it's known that reinstating DARS1 enzyme, even to a low threshold, may actually help to improve or even prevent disease. And then the efficacy of treatments to rescue the HBSL pathology is actively being investigated in the mouse, iPSC, and organoid models. And then finally, again, no central registry or ongoing natural history study specific to HBSL. This is something that we think is really more immediately actionable. So we need to understand, to do this, to expand our knowledge of the phenotypic spectrum, and we know actually where most patients are distributed, which is between 5 registries at these institutions listed. So this is something that we really could and should start obtaining, reviewing, curating, and centralizing a patient database. And then finally, we do hope to potentially leverage the existing LBSL experience, infrastructure, and tools to quickly make some advances on this goal. Okay. So thanks very much, and with that, I'll hand it over to Beth McGinn, who'll talk more about LBSL successes.
The microphone's not working. Sorry, yikes. Okay, can you guys hear me? Okay. My name's Beth McGinn. My husband and I founded A Cure for Ellie in 2013. It's named after our daughter who was diagnosed with LBSL at age 3. And Ellie is now 14 years old. And so as she has matured and become a high schooler who doesn't really want any attention on her, our organization has matured along with her. And over the course of a decade, we've connected with families all across the world. And so we're transitioning to cure LBSL in the next month. And when Dr. Smith-Fine said we were ultra rare, we were definitely ultra rare. I think we're less than 200 patients in the world. Although that number could be higher just from patient-to-patient communication through our foundation. We do know there's a higher than normal prevalence in Russia, but there's not good data coming out of there. So that number could be higher, but regardless, we're still ultra, ultra rare. And we are the only organization in this space. We created it to fill a void, and that's both good and bad. It's good in that we're very streamlined. The patients are all in touch with us. We all come to the same conclusions and can move forward very quickly. But it's also bad because we're the only ones doing this, right? So, I like to think of us as small but mighty, and the patients who connect with us over Facebook, we have about 150 families that connect with us through Facebook, are really, really engaged. And together, we have raised $300 million toward research, which for such a small group of people, I think is, I'm pretty proud of us. And we've done it through, lemonade stands, 5K races, that sort of thing. So very, very engaged group of individuals, which I think is one of the strengths of our community. I think that's one of the reasons why on the heat map, we trended a little bit more toward the green side. So we've held 3 international patient conferences, which have been each been a success. We do them every 2 years, and we do them both virtually and in person, and we usually have about 100 people who participate in those. And then we have usually over a dozen scientists and doctors who come together as well. As mentioned before, we just got an awesome grant from the Chan Zuckerberg Initiative, which is really kicking our patient advocacy into high gear. So we have been very fortunate to be able to hire a director of patient engagement who's here today, Melody. Thank you for being here. And so in the next 12 months, we hope to launch a contact registry, and then that will be the basis for eventually a patient registry, which we would like to use to help amplify the work that Dr. Amena Smith-Fine is doing here in the United States with her natural history study. So we as a patient organization hope to be able to help her collect data and make sure that that data is useful data and that it is linked up with what she's capturing so we're not rushing into that. That's going to take some time, but thanks to the initiative, the grant we received, it is it's feasible for us. So we're undergoing a name change. We're going to have our registry in 22 languages. And going back one, I think you can see, this is based on our Facebook connections, but you can see where our patients are. We seem to have a lot of patients highly engaged in Brazil, Finland. There's a higher than normal prevalence as well. So we're going to translate our contact registry and our eventually our patient registry into 22 languages. And so another huge success, and I feel like Dr. Smith-Fine should be talking about this because this is her baby, but we've been really, really excited to participate in the natural history studies over the last 4 years. This is my daughter, Ellie. And when we launched this 4 years ago, WebMD actually came and did a story on it because it was so novel and so cool that we didn't have to travel to the hospital to do these studies. They send us the package in the mail, we open it up, we turn on the laptop, we see Dr. Smith-Fine, she gives my daughter instructions, and all of that data about sway and gait is immediately transferred back to her lab. So it is still pretty cool and pretty novel. And from a patient perspective, it's wonderful because we don't have to get on a plane to participate in research. And we don't have to expose my daughter to germs unnecessarily. And this was all pre-COVID. So we are expanding it. It's actually Dr. Van de Knap, who's here, and Dr. Englund have been replicating this in the Netherlands. We're very grateful for that. And then we just recently, thanks to help from our patient community, identified researchers in both Finland and Brazil who are interested in replicating the studies there. So very, very excited about the future of that. I think another reason we trended green was because we've made a lot of connections that have been helpful. And so we're connected with a lot of umbrella organizations, even though we're so little. And some of them are here in the room, like United Leukodystrophy Foundation. And so that was another criteria on the rubric of our engagement. I think that kind of helped us. And then connecting with LBSL researchers, thanks to Melody's work, she went through every single paper that has ever been published on LBSL, and she created a database of 300 doctors, clinicians, and researchers who have in any way been associated with our disease ever. And so we're very excited once we switch over to Cure LBSL officially, we're gonna be reaching out to everybody and hopefully identifying more patients that way, and then laying the groundwork for, one day if we get to clinical trial, we wanna know where all our patients and all our doctors are. So this is where we are on our timeline, as you can see. Also, FDA engagement was another category on the rubric, and we are next year planning to do a listening session with the FDA. First, we need to kind of capture the patient story, the disease burden, and so Melody's helping us with that. We're going to start collecting stories. Infantile onset, as mentioned earlier, is more severe. This young boy is Tasos. He's from Greece. He's 4 years old now. He has microcephaly. He's obviously severely impacted, and he's a very sweet little boy. So we need to capture the disease burden, not just for people like my daughter who go to school and walk around in the house every day, but for kids like Tasos. We need to kind of understand what life is like for his parents. So that's what we're busy doing in the next year and a half. So I think that was it. I think that's where we're at.
Hi, thank you. I think I'm next. So my name is Christina Nemeth Mertz. I'm an assistant professor at the Kennedy Krieger Institute in Baltimore, Maryland, with Dr. Smith-Fine. So I'll be talking about the specific challenges for LBSL, and I really kind of enjoyed hearing sort of Beth's stories. A lot of the challenges that we have, she spins into these positive things, and I mean, there's such an overlap here, but I mean, I think, I have such a positive outlook with this, because I think many of our challenges are sort of somewhat addressable, and hopefully we can get there together. But the main challenge in LBSL revolves around the fact that it's an ultra-rare disease. So I have a little schematic here that's going to be growing, but it's basically to drive the point home that every challenge we face is because we know of so few patients. And as Amena mentioned, there's probably 200 worldwide that we know of. Generally, the carrier rates are very low. As they both mentioned, within the Finnish population, there's a carrier rate of about 1 to 95. That seems to translate really well in the Russian population as well. We've had interaction with those patients during our conferences. And as she mentioned, we just don't have the data. So likely there are many other patients. If we can identify these patients, obviously that's going to help us with a lot of our other endpoints. The rarity certainly links to limitations in the natural history study. So these studies lack rigor, of course. We have difficulty to find, recruit, and enroll these patients to get robust studies. And a lot of that has to do with diagnosis. So the map that Beth had of the world, I think, is really reflective of this fact that access and affordability of genetic testing and imaging is really limited in many places, and that is likely what results, or at least contributes to some of the underdiagnosis and misdiagnosis. We've had interaction with many adult patients who, spent many years of their life being told they had something else until receiving a diagnosis of DARS2, and we're sure that is not sort of a rare experience. Another issue is that in many parts of the world, including parts of this country too, most likely, clinicians may not be aware or experienced with LBSL. That's fine. It's a very rare disorder, but again, that's going to contribute to those low numbers of patients. Lack of biomarkers. Of course, finding a biomarker that's sort of easily accessible rather than having genetic screening and MRI would facilitate diagnosis and tracking of disease severity. That, of course, again, is related to sort of the rare prevalence of this disorder. And of course, early diagnosis is a challenge, and as we've mentioned a few times now, newborn screening is not feasible related to many of these factors. Continued challenges, the need for increased patient research participation internationally. That's going to help to really define a disease mechanism. It's critical for developing disease-modifying therapies, of course. And then, again, the lack of guidelines for care that can be published as an international standard. So, as you can see with my schematic, we have arrows all over the place. But again, this is sort of just a very basic challenge is the rarity, and then all these other factors are related to that. And the last thing that I'm going to touch on is sort of the modeling. So we have a number of different models that have been used for about the last 15 years. So the last 10 years has really been a time of growth for LBSL research, and a lot of that comes from Dr. Vanderknap's group. The models, as we've mentioned, do not recapitulate the patient condition perfectly. However, I like to think that the models that we have sort of have contributed to our knowledge and our understanding of LBSL and certainly of DARS2 functioning. Much of the work has been patient lymphoblasts, fibroblasts. Of course, these are sort of accessible cell types. A lot of it's cell constructs and patient-derived iPSCs and other cell types, which I'll talk about. And again, as none of these models really accurately can tell us what's going on in the patient, we have garnered a lot of useful information, and I have a not exhaustive collection of data here, but I just wanted to point out these are viral constructs that researchers have used to understand the subcellular localization of DARS2 in normal conditions and, of course, with different patient mutations. We've learned a little bit about immunoisolation of the recombinant proteins using E. coli. And of course, these studies would have to be repeated when we have sort of better models, but I think this is a really good way to understand how immunoisolation is affected by different mutations. And then using different constructs in HEK cells as well as other tumor cell types, we've learned a lot about splicing deficits, and these are cell type-specific splicing deficits that we believe to play a very large role in the progression and the pathogenesis of LBSL. Our group has done a lot of work towards iPSC-derived models, so a lot of this has come through A Cure for Ellie. So through that foundation, we've been able to access patients, we've been able to collect samples and turn PBMC blood samples into iPSCs, and then through the help of other researchers, derive these iPSCs into neurons, motor neurons, which we can study. And from these studies, which are in the process of being published now, we've learned a lot about mitochondrial function. We can study growth of these cells. We can study the arborization. We can study electrical activity and understand sort of the neuronal deficits in the dish from different LBSL patients. Of course, one limitation of this kind of harps back to what I mentioned before, is that we only have a limited number of lines. There are many, many different mutations that are associated with patients with LBSL. Patients have compound heterozygote mutations, so of course we don't fully understand the combination of these mutations and how that affects the function of the neurons or how that affects the progression in the patient. However, I think these cell experiments are sort of a first step towards making those gains in knowledge. And then other work from our group has been using iPSC-derived cerebral organoids. So we use an unguided protocol to derive these organoids. We study them for many, many days. We've grown them up to over 100 days. And then one of the most recent studies that we did is doing single-cell RNA-seq to study the cell type-specific effects in these organoids. So when these organoids are about 70 days of age, which we found to show sort of the most diverse array of cell types, we did a very high resolution screening able to understand a little bit more about the transcriptomics, how specific mutations contribute to the splicing deficits, the cell type specificity of those splicing deficits, and then how different mutations may contribute to a unique gene signature in these cells. And so this is also work that we're working on publishing right now. And finally, I want to touch on the mouse models. So again, these models don't accurately recapitulate the human disease, and the main problem there, well, one of the problems is that complete knockout of DARS2 is embryonic lethal. This is not unique to DARS2. This happens with basically any of the tRNA synthetases, so these are essential for life in the cytosol or in the mitochondria. We have also attempted to do nestin-specific, so knocking out this gene within CNS-specific cells is also embryonic lethal. And because of that, we've been sort of limited to conditional knockout models. This is a mouse that was developed in Cologne, Germany by Dr. Sandra Trifunovich's group, in which they designed a LOX-P model in which the third exon of DARS2 was localized and using Krieger recombination removed from cell types, from specific cell types. And the reason that's the third exon is that is sort of the splicing deficit that we most commonly see in 90% of the patients. So these patients lack exon 3 in DARS2. That leads to a truncated protein and a loss of that protein. And so that's sort of what they were going for, the design in these mice. We and others have used these mice to study what happens when DARS2 is removed from specific tissues. And we see mitochondrial effects in the heart, in the skeletal muscle, and then we have done that in the brain, studying CAMK2 specific neurons in the hippocampus and the forebrain. Dr. Trifunovich's group has also sort of expanded on this. I might have this on the next slide, in the cerebellum. And what we understand with these models is that deleting DARS2 from specific cell types is detrimental to neurons. It leads to a progressive loss of those cells, and you can also see in the first figure here, that leads to mitochondrial deficits, specifically in the neurons that are affected. And one of the most interesting sort of aspects to me that comes out of this is if you repeat the same process and target oligodendrocyte producing or oligodendrocytes, whether they be oligodendrocyte precursors or mature oligodendrocytes, which is what this graph is showing in the MYKO, that's the myelin knockout, we do not see a phenotype in those cells. So this seems to be, at least in the mouse, very specific to neuronal cell types. So again, the mice are not perfect. We do need to spend, or there needs to be sort of a concerted effort in finding these system-wide models that would better recapitulate the patient condition. We have not targeted the spinal cord. Obviously, the spinal cord is the most affected in these patients, and so that's something that we're looking into. And one thing that I forgot to mention that I will mention is that attempts have been made to generate mice with human deletions, and for whatever reason that has not been possible. That site has not been amenable to editing. I say that here, DARS2 gene shows resistance to point editing, and that's sort of where we are with that. But alas, we will continue to try. So I think that's all that I have, and I think we can take some time for questions.
Excellent. Fantastic work. I'm so excited about you getting a Chan Zuckerberg Foundation grant, so that's congrats. Questions?
Can you hear me? Yes. So I have a question. I love the sort of efforts in the patient registry and the multi-site natural history study. Given I'm going to just speak loudly. I was wondering if you've made efforts to align those, right? And if so, how you're overcoming regulatory concerns, data privacy concerns, or multi-nation sources, and how you're, because I think that's such an important model for this whole group. How we work, registries in multiple.
Just maybe repeat it, Amena.
Yes, so it sounds like from what I could gather the question, she's asking about the natural history studies and the patient registries that are multi-site, and if those efforts have been aligned. I guess that was one, first of all, have they been aligned, and sort of asking a little bit more logistics about how we've done that or what we've learned. And I'll talk about the natural history study. Beth can talk a little bit more about the plans for the registry, the LBSL specific registry, which hasn't quite started yet. But I guess, well, it's a small community of investigators who are focused on LBSL but are very passionate with it. And so, I mean, in our case, like a few years ago at like one of the previous in-person ULF meetings, our group at Kennedy Krieger met up with Dr. Mario Vandernap and Mark England, and basically we just sort of established a connection at that meeting and said, hey, we decided we were interested in trying to work together on this initiative, and there's history with other leukodystrophies, primarily ALD there. So we just were very intentional about it in terms of planning some meetings, and we essentially just shared our protocol directly, and they developed really a parallel protocol. And then we have a data sharing agreement, and we trained them remotely because this was all in the midst of the pandemic. And that's the same model that we're using in Finland and that we hope to reproduce. Yeah.
In order to submit for regulatory purposes, you need source verified data that is effectively identified. So how are you, or have you already considered getting across the multi-site source verified, identified, subject level identification, joint submission of that data?
That's a great question. Practically, we haven't gotten to that step. Sorry, I misunderstood the question. Yep.
And so the same question goes to like the collaborations with the patient registry. How are you dealing with patient identification? But you're such an ultra-rare disorder.
I think yesterday the patient groups met, and we learned so much. And I think that's the beauty of this meeting. So thank you so much for hosting us. We realized, I think just yesterday, that we need to take this slow and we need to do it right. And we need to make sure that the data that we collect is the data that's going to be useful in terms of getting drug approval. And so, I mean, we're still, the answer is we're still figuring it out, but because of this meeting, we at least have more sense of what we need to do. So, thank you.
I have the same problem. I was just wondering if you intelligently figured it all out.
When I do, I'll call you.
Hi. Thank you for your. And we already talked about this in the group, but I'm concerned about disease models that don't really replicate the disease and what you can conclude from them, because patients never have two null mutations. So if you have a knockout, even if it's a cell-specific knockout, then I'm very concerned that it tells something about the pathogenesis of the disease.
Okay. I mean, I think that's a very... Yes, yep. So Dr. Vanderknap expressed concern about the modeling for LBSL and the fact that they do not recapitulate the patient condition, and as a result, how useful the information is that we gather from those models. Is that correct? And I agree. I mean, I think we have to try. I think getting sort of any information about the gene function, gene interactions. I think all of these sort of aspects of data collection from different models, whether it be mouse models, cell models, I think all of these, in my head, go into a large pot where we mix it all up and then see what we can sort of pull out. And I think the point is to get all the information we can. None of this stuff is going to go directly from mice into people. This is, going to be a long process of understanding sort of gene interaction, treatment strategies, how the gene responds, how the animal responds, how cell models respond to different therapies, and then sort of moving slowly and very carefully forward into people. Eventually, obviously, that's the goal, but no one looks at these models and thinks that they have LBSL. It's a matter of taking what we can from them and working very slowly and diligently to basically make the best decision. So I'm hopeful that some of these mouse models and certainly some of the cell work is going to lead to a better understanding of DARS2 functioning, and we just take that information and move slowly. I hope that, that's all we can do, I think.
Great. Maybe just in the interest of time, we'll wrap up so we move to the last session, but this was terrific. Thanks to all the speakers.